slide. So we begin with methionine, and methionine can come from a dietary source like protein. Remember that it is an essential amino acid, and methionine is special in that it is one of only a few amino acids that contains sulfur in its structure. So if we want to start metabolizing methionine, we need an enzyme known as methionine adenylyl transferase. And this enzyme, what it essentially does is what its name suggests. It transfers an adenosine group onto methionine, and that adenosine group actually comes from ATP, or adenosine triphosphate. So methionine adenylyl transferase takes the adenosine group from ATP, adds it to methionine to form S-adenosyl methionine, or SAM. So here is the adenosine group right here. So this leads to this important molecule known as S-adenosyl methionine, or SAM. So SAM is a very important molecule in that it becomes very important as a methyl donor. So as you see here, the methyl group in methionine and an S-adenosyl methionine are the same. So what happens is this adenosine group that gets added to the sulfur of methionine makes the sulfur and methyl bond unstable, allowing SAM, or S-adenosyl methionine, to become a methyl donor. So that's what its purpose becomes. So SAM is involved in the synthesis of catecholamines, nucleotides, melatonin, etc. We'll talk about this in another lesson, but just remember that S-adenosyl methionine acts as a methyl donor for the synthesis of a variety of different things in our body, including catecholamines, nucleotides, creatine, and melatonin. So the next thing we want to do in methionine metabolism is we want to process SAM by methyltransferase enzyme. And what this essentially does is it removes the methyl group, it adds it to another molecule. Those molecules could be part of catecholamine synthesis, nucleotide synthesis, etc. And this leaves us with S-adenosyl homocysteine. So it's no longer, the methyl group is no longer attached to this sulfur group now. After that, S-adenosyl homocysteine can then be acted on by adenosyl homocysteines. So what this does is it actually removes the adenosine group from homocysteine, leaving us with simply homocysteine. And homocysteine can then be acted on by an enzyme known as methionine synthase. And methionine synthase actually regenerates methionine from homocysteine, and in the process it actually takes a 5-methyl tetrahydrofolate, or THF, and converts that 5-methyl tetrahydrofolate. It removes the 5-methyl group from tetrahydrofolate, and it forms tetrahydrofolate in this process. So this process constitutes a cycle, and this cycle is actually known as the activated methyl cycle. This is what we utilize in catecholamine synthesis, as I mentioned before, for when we need S-adenosyl methionine. But what's also important about this cycle is that it actually generates tetrahydrofolate, or THF, and this is important in a variety of processes as well. So this portion of the pathway is known as the activated methyl cycle, and it is important to form two things. It's important to form S-adenosyl methionine, which can be used as a methyl donor for various processes, and it's also important in generating tetrahydrofolate, which is used in a variety of processes as well. And importantly, with regards to methionine synthase, it is one of the 2 enzymes in humans that requires vitamin B12. So again, this is extremely important for recycling of homocysteine into methionine. We need vitamin B12 for methionine synthase. Now, if our purpose is not